Hello, and welcome back to Introduction to Genetics and Evolution. In this set of videos, we'll be getting to the Hardy-Weinberg equilibrium. This is a foundational concept for the field of population. Before we go to that, however, let's talk about allele frequencies and genotype frequencies, because understanding what those are and how you get them is very important both for the Hardy-Weinberg equilibrium and for population genetics more broadly. Now let me ask a couple of broader questions first. First, can we predict genotype frequencies if we know allele frequencies? If we know how abundant different genotypes like big A, big A, big A, and little a are in a population, can we get from this to allele frequency? And if the answer is sometimes, when can we? The other question is, do genotype frequencies intrinsically change over time, or do they remain constant? Let's assume, for example, that big A is dominant over little a. Do we expect over time big A's to become more common just because of dominance? Well, we'll answer these by looking at variation one gene at a time. We're not going to be looking at interactions among genes here at first. So let's take this hypothetical scenario for the A gene. Let's say this A gene has two alleles, big A and little a. And let's say there's three possible genotypes, of course, from these two alleles, big A, big A, little a, and little a, little a. Well, by definition, frequencies have to add up to 100%. If there's two possibilities, as with the alleles, if there's a big A and a little a, the total of the fraction of little a plus the fraction of big A must be 100%. So if we have 78% big A, then we necessarily must have 22% little a. Same thing happens with the genotypes, that if 25% of individuals are big A, 50% are big A, little a, the only other possibility is little a, and therefore that must also be 25%, making the total 100%. So how do we calculate genotype frequency? Well, we can definitely calculate genotype frequencies always when we have the counts of the different genotypes. So let's assume in this case that every individual is a diploid. They have two copies of all their genes. So we can literally just get this by counting. So here's a population. There's big A's, big A, little a's, and little a, little a's. There are 10 individuals in this population. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. The frequency of big A will be its abundance, which there's only one right here. So it would be 1 out of 10, or 0.1, which is the same as 10%. Frequency of big A, little a's, there are 1, 2, 3, 4, 5, 6 out of 10, or 0.6. For little a's, the ones that are not circled, it's 3 out of 10, it's 0.3. So you add up these three numbers, and again, it adds up to 100%, as we expect. What about allele frequencies? Well, there's two ways we can actually do this. The most obvious way, which is not necessarily the better way, is to literally just count the big A's and the little a's in this group. So we can look at this and see, well, there's 20 total alleles here. They're attached together as genotypes, but there are 20 alleles. How many big A's are there? 1, 2, 3, 4, 5, 6, 7, 8. 8 big A's out of 20. So how many little a's are there? There obviously must be 12. So we have 8 out of 20, or 0.4 big A's, 12 out of 20, or 0.6 little a's. And again, this adds up to 100%. The better way, when we're working with a large number of genotypes, rather than counting the individual alleles like this, is to look at the genotype frequencies. We can say, we know that big A's are completely made up of the big A allele. So the frequency of big A would be the frequency of big A's, and half the frequency of big A, little a. So these are the numbers we used from the previous slide. Remember, our frequency of big A was 0.1. Our frequency of big A, little a is 0.6. This is from the previous slide. We add up 0.1 plus half of 0.6, which would be 0.3, comes to 0.4, which is exactly the same as what we saw up here. Similarly, we can say the frequency of little a, or 0.3 plus half of 0.6. So that's 0.3 plus 0.3, and that is 0.6. That's exactly what we see over here. And again, these add up to 100%. This is what we expect. Now, you can think of the world as basically a pool of gametes. It's not a particularly pleasant thought, but you can think of it that way. All individuals of sexual species start as two gametes, right? That you at one point in time were two different gametes that came together and fused into you. Now, gametes are one end. They just have one copy of all their genes. And this vision is not as artificial as it sounds if you just imagine, as it shows here in this picture, all the gametes floating around, because many marine invertebrate species spew their gametes into the water, and those are the ones that ultimately come together to make individuals. In this picture here, about 60% of the gametes are big A, 40% of the gametes are little a. And what we can do to estimate, if we assume that these gametes just come together completely randomly, we can use a joint probability multiplication to figure out what the genotype frequencies will be in that next generation, the people who are the individuals who came together from the gametes. So if 60% of the sperm are big A, 60% of the eggs are big A, so we're just saying 60% of all the gametes, 40% of the sperm are little a, 40% of the eggs are little a, what would the probability of a big A individual be? Well, probability of a big A individual would be the joint probability that an A sperm comes together with an A egg. Well, A sperm is 0.6. A egg is also 0.6. So that would be 0.36, or 36%. We can do this for all possible individuals. So here's, 0.6 times 0.6 is 0.36. For little a, it's 0.4 times 0.4, so that's 0.16. But there's two different ways you can make a heterozygote, right? You can have a big A egg and a little a sperm, or a little a egg and a big A sperm. So each of those is 0.6 times 0.4, 0.6 times 0.4, or 0.24 twice. That comes out to 0.48. And again, as we expect, we can add all these up. It comes out to 1.0, or 100%. So we're assuming here that the gametes just come together completely randomly based on their proportions in the population, which is an assumption we'll come back to, and that's relevant to Hardy-Weinberg. Now let me show you just another way of visualizing the same thing. Rather than using this sort of joint probability approach, let's visualize it a little bit differently. We can use a modified Punnett square to show the relative amounts of the big A and little a gametes. So again, this is exactly the same thing. It's 0.6 big A, 0.4 little a. And we have here the sperm on this axis, the eggs on this axis. Note the size is here, the relative size. This is doing a 6x6 block. This is a 4x6 block, 4x6 block, and 4x4 block. We can use that then to estimate what we'd see in terms of the genotypes. And doing that, we see exactly the same results that we saw before. That here we have 36% of all these blocks, or 36 out of 100 blocks are big A, 16%, or 16 out of 100 are little a, 24 plus 24 is 48% big A, little a. Now, can we calculate the allele frequencies from these genotype frequencies? Well, the answer is yes. We can always calculate allele frequencies when we have the genotype frequency. Because again, we know all the big A's have big A. They are all big A, so 0.36% is that. Big A little a's are half big A, so half of 0.48. So 0.36 plus 1/2 of 0.8 is 0.6. So here in the offspring, just like in the gametes that formed them, the frequency of big A is 0.6. Frequency of little a has to add up to 100%, so this must be 0.4. Okay? So we see that not only, so we have the frequencies of the alleles being 0.6 and 0.4, the genotypes being 36%, 16%, and 48%. And then in the next generation, if they were to spew out their alleles again, the allele frequencies are still the same. Since the allele frequencies are still the same, this is all a self-perpetuating process. That the allele frequency was 0.6 in the gametes, it created these genotype frequencies, and these genotypes will produce 0.6 frequency of big A gametes. So this is a process that is basically stable with these assumptions. Now the pattern that you see does have a wide variety of assumptions, but the stability is what's referred to as the Hardy-Weinberg equilibrium. We'll come back to this more in the next video. Thank you.